hear me and see the screen?
Yes.
Great, thank you very much. Good evening, good morning, good afternoon. I'm not sure what to say. I do want to begin by thanking the organizing committee for this very kind invitation and the opportunity to talk about our work, which we've been doing in Huntington's disease. I'm going to come from left field a little bit because what I'm hoping to do today is not to talk about the potential of cell replacement therapy in Huntington's disease, although I do believe in the approach for treating other neurodegenerative disorders like Parkinson's disease, as we've just elegantly been shown. But what I want to do is to show you the work that we've done in the last decade on transplantation studies in animal models and clinical trials, to see what we've learned about disease mechanisms and the tangent that we've taken, gaining a better understanding of the disease to develop and identify new therapeutic approaches to treat this disorder. So for those of you who are perhaps less familiar with this, I'm going to do a little bit of a recap on Huntington's disease. It is a genetic disorder of autosomal genetic transmission. So when the father or the mother is a gene carrier, it is expected that half the progeny will develop the disorder in their 40s or 50s. This is due to an expansion of a CAG repeat in exon 1 of the Huntington gene. The more or the larger the expansion, the more severe the symptoms will be, and the earlier the patients will develop the disease. So there is a juvenile form of the disorder, but normally the patients have around 40 to 42 CAG expansion. And what happens in this disorder is that patients have very striking motor abnormalities, specifically chorea, which are these very large involuntary movements. But as the disease progresses, the patients are actually bedbound, so they become very rigid and incapacitated. What you don't see on the video is that these patients also suffer from very significant cognitive and psychiatric impairments. Of course, there is an anatomical substrate to this, and it's the fact that as the disease advances, the patients have or are characterized by a very striking degeneration of the basal ganglia. So you can see here that as you progress to a more severe HD phenotype, these structures are majorly atrophied because we lose a lot of the cells that project different structures. Another hallmark of the disease is the fact that the gene mutation will cause the expression of the mutant form of the Huntington protein. Huntington is very important in synaptic transmission, is sort of a scaffolding protein. So when this mutation starts to occur, you'll see these aggregates found in the cytoplasm and the nucleus of various cells, and in fact, in every cell of the body, because this is driven by a genetic mutation. Here you see a picture of these cortical inclusions in an early stage HD patient. Now, a few years ago, almost a decade ago, my lab was tasked to do the post-mortem analysis of the very few patients in the world who had received cell replacement therapy in the hope to slow down or even cure their disease. And what we were sent were the brains of 5 patients who had entered a clinical trial in the US. These are very small pilot studies, and 3 brains from patients who had entered a clinical trial in the UK. Now, these 2 trials have a difference in the way that the clinicians approached it. In one case, these are all fetal tissue. So in the US trial, the surgeons dissected a little piece of tissue from the fetus that originates or yields striatal neurons, and they implanted this piece of tissue in the brain. And you can see by the dotted line that it's very easy to delineate the anatomical border of these grafts. And within these grafts, you find 2 distinct zones that we call P zones and non-P zones. The P zones are areas that are very rich in striatal neurons, which are the neurons that we're trying to replace. In the UK trial, the clinicians opted to mechanically dissociate the cells and retransplant them into the brain. So that's why you see individual cells in the transplant. Now, we studied these brains because the patients did not really benefit from cell transplantation, and they had very, very little, if any, clinical benefit. So we wanted to understand why this was the case. And we studied these transplants for a number of years. But one of the most striking observations that we made is that we found the genetic product of Huntington's disease, the mutant Huntington protein, in the transplants. So we found these little inclusions of MHTT both in the solid pieces of tissue, in the P zones and the non-P zones, but also inside transplanted cells, individual cells, and even in the neurites, so the projections of these neurons. Now, this was pretty striking to us at the time because it meant that the diseased brain was affecting the normal tissue that had been transplanted in these brains. And it had, of course, implications for treating these patients with cell replacement therapy. And one of the first idea that we had, because we had seen them in the neuropil and in these neurites, we thought that there was a possibility that the mutant protein could propagate between the diseased brain and the normal tissue. So we investigated this with a colleague probably of yours, Ji-won Song in Korea, who actually elegantly tried to investigate this by transplanting wild-type animals at postnatal day 2, intraventricularly with fibroblasts derived from HD patients with a very severe mutation. Remember, I told you that above 39, you are inclined to develop the disease, but 143 CAG repeat means that this was an individual with juvenile case of Huntington's disease, so a very severe mutation. And what Ji-won and his group did is that they did behavioral assessment over time and then sacrificed the animals that they sent to us. At 30 weeks post-transplantation, they actually started to notice a change in the behavior of the animals. So the first thing that they did is that they did a clasping test, which is a classic test for neurological dysfunction in animal models of Huntington's disease. And you will see here on the top panel, an animal that has been injected with normal fibroblasts, and at the bottom, an animal that has been injected with fibroblasts derived from a Huntington patient. And you can see that the animal at the bottom is starting to do this clasping, typical phenotype of Huntington's disease in mice. They also looked at cognitive impairments because we know there's a component of cognition in Huntington's disease, and they did a simple swim test where you will see on the left animals injected with normal fibroblasts, which find the platform very easily after training, but an animal who's been injected with fibroblasts derived from a Huntington's patient is really struggling to find the platform, even though they've been trained to do so, which indicates, of course, that they have some kind of cognitive impairments. When we received the brain, we started the postmortem analysis, and what we noticed is that despite the fact that the cells were injected into the ventricle, we found very few cells over time because we sacrificed the animals at different time points within the brain of the animals. And this is probably because the animals were not immunosuppressed. We injected human cells and we did not give them an immunosuppressant. So eventually probably the cells died of an immune response. But as they died, they left behind mutant Huntington protein aggregates, especially in the striatum and in the cortex, which was accompanied by an increasing microglial response. Now, these are all neuropathological features of Huntington's disease. So in other words, we were able to recapitulate all features of Huntington's disease, both behaviorally and neuropathologically in completely normal animals if we injected them with cells that were derived from Huntington patients. So I want to tell you this is very important because all these years we've thought that Huntington was strictly caused by the mutation and you needed the mutation to develop the disease. But what this is showing is that by being exposed to cells that carry the mutation alone, you can develop a full-blown phenotype that relates to Huntington's disease. We then went on to do the same experiment with extracellular vesicles derived from the fibroblasts. So we took the fibroblasts, stimulated them in vitro, isolated the extracellular vesicles that we injected again into the ventricles of neonatal wild-type animals, did the behavior, sacrificed the animals. And this time what we saw is that we had the same problems with the locomotion and the cognitive impairments, but this time much earlier than 30 weeks post-transplantation. We started to see these impairments at 12 weeks post-transplantation. So we accelerated the process of disease manifestation with the vesicles, which implies that the vesicles are an extremely good vehicle of disease propagation, even more so than the fibroblasts themselves. So having established this, we became extremely interested in understanding the routes by which these proteins could propagate and what could be done to actually stop this from a therapeutic point of view. So we developed a model of parabiosis in the lab, which consists of fusing two animals together by their flanks. So we do surgery, we suture them together. The blood vessels start to fuse together within a few days. So then the circulation of the HD mouse starts to feed the wild type mouse, and the blood of the wild type mouse now circulates within the HD model. We're able to keep these animals for up to a year. So we did several pairs of Huntington mice with the wild type mouse, but we also did Huntington mouse with a Huntington mouse and a wild type mouse with a wild type mouse as a control. And then we took blood samples every 3 weeks, and we sacrificed the animals at different time points. The first thing that we looked at was to make sure that the parabiosis technique worked. So we looked at erythrocytes within the wild type animals paired with an HD animal, and we found out that all the red blood cells with mutant Huntington protein within them that you normally found in the HD mouse were also found in the wild type mouse. We looked at plasma, and we found exactly the same thing. So the wild type animals expressed mutant Huntington protein within their plasma. So we were able to show or demonstrate that the fusion of the two animals made the circulation of the HD blood into the wild type animal. Then we looked into the peripheral organs, for example, the liver, at 6 months post-parabiosis. And what we saw is that the HD mouse paired to a wild type, we know that it expressed a lot of MHTT, because I told you before, because of the genetic mutation, the mutant protein is expressed in every cell of the body. But when we looked into the wild type animal paired to an HD, we were also able to find mutant Huntington protein in cells of the liver. Rare occasion, but it was still there. We found this in the kidney, in the muscle, in every organ that we looked at. We also looked into the central nervous system. This is a picture of all the inclusions that you normally see in the neurons of an HD animal. But when we looked in the wild type, we also found these inclusions in neurons of the wild type animal. And in fact, when we ran a filter trap assay, we were able to quantify the quantities of aggregates and concentration of protein, and found that there was a greater number in these wild type animals that were paired to an HD animal in comparison to wild type. Now, this is very important because remember, this is blood circulating in the animal, and we're able to reach the central nervous system and have an effect that recapitulates some aspects of Huntington's disease in a completely normal animal. Now, one thing we did not expect, and naively we did not think of, we were most interested in looking at propagation of the protein and its effect, if we could recreate aspects of the disease. But we actually saw that the wild type blood could dilute and attenuate the pathology of Huntington's disease. So again, we looked in the liver. This is the picture I showed you before, a Huntington mouse paired to a wild type. You see these inclusions within cells of the liver. If you look at a Huntington mouse paired to a Huntington mouse, you see a lot of these. And if you see a Huntington mouse that's not paired with anyone, then you see a lot of these, but you see a lot less in the animal that's paired to a wild type, and we can quantify this, and you see that it's significantly different. So we can, I would say, dilute or temper the MHTT load in the peripheral organs if we keep the animal parabios together for 9 months. We also looked at cytochrome C, which is a measure of mitochondrial dysfunction. And we were able to see improvement of mitochondrial dysfunction in the kidney, the liver, the muscle, 9 months post-parabiosis. In other words, the good blood does not only eliminate some of these proteins, but it actually improves cell function peripherally. We saw similar effect in the central nervous system. Here we looked at markers of cell populations in the cortex or projection neurons in the striatum, and you can see that we have an improved profile in the HD mouse that receives continuous blood supply from a wild-type mouse. Now, in other words, we're, by joining these two animals together and providing either diseased blood or good blood, we're able to show propagation of the mutant protein in a system, not only peripherally, but into the central nervous system. But most importantly, we're able to dampen or attenuate these phenotype and neuropathological features when the HD animal is supplied with wild-type blood. So we have two theories about this that we think is very important, because now we became very interested in the crosstalk between the periphery and the central nervous system. And the first theory that we have is the following. Now, we've analyzed the blood samples of many patients with Huntington's disease. We know that every cell of the blood expresses mutant Huntington protein or oligomers or aggregates. From a previous study where we have shown that the blood-brain barrier, especially the endothelial cells, express mutant Huntington protein, which seems to impair their function, it makes the barrier more permeable. So the blood cells, let's say the white blood cells, are more easily transmigrating into the parenchyma. And because there's lots of inflammation in these brains of neurodegenerative disorders, for example, a white blood cell will transform into a macrophage and eventually die. And once it dies, it will leave behind all of these aggregates or oligomers, which are toxic forms of the protein, into the extracellular space, which will be able to be picked up by other types of cells like microglia or other neurons. The other possibility, as shown also with the group of Ji-Won Song, is that under stress or inflammation, cells of the blood will start to shed microvesicles. Microvesicles are small clones of the cells. They contain DNA, RNA, viruses, proteins, everything from the cell. And because they're so tiny, you don't need breaches or increased permeability of the blood-brain barrier. They can cross easily into the parenchyma. They will eventually disintegrate, and they will leave behind pieces of proteins that can be picked up by other cells. So having been interested in this now, we have developed a fully humanized 3D blood-brain barrier model based on iPSCs. We have developed them for control blood-brain barrier, Huntington barrier, Parkinson's disease barrier, idiopathic, genetic forms, etc. We use a chip from Imetas that we continuously perfuse. We differentiate all cells into a layer of endothelial cells, a matrix, astrocytes, and pericytes. These barriers look like this. They are fully formed. They're leak tight. If you inject them with different fluorochrome like dextran or rhodamine, they do not leak at all. And in fact, we have started to use them to study, let's say, the transport of vesicles, pathological protein, etc. This is an example of the barrier with a layer of extracellular vesicles that we've exposed the barrier to. You can see that the vesicles enter the endothelial cells, or they can cross all the way across the barrier. And in fact, now we have seeded neuron-derived, iPSCs neuron-derived on the other side, so we can study the effect of this on the central nervous system components. Now, I've talked to you a lot about the propagation of mutant Huntington protein, but one thing I have less talked about is the seeding capacity as well. So it seems that these proteins that are associated with neurodegenerative disorders are also able to act like a prion and actually recruit the endogenous protein to transform its behavior. So here I'm going to show just a tiny bit of data. I have a lot more, but I'm just going to show you the in vivo paradigm where we demonstrated the seeding capacity of MHTT. And what we did is that we had a collaborator in France, Ronald Melky, develop these recombinant synthetic human fibrils. This fibril is normal because the CAG repeat is under 25, but this is an abnormal fibril because the CAG repeat is above 48. And what we did is that we injected these fibrils in the ventricles of neonatal mouse models of Huntington's disease, the R62 mouse. The R62 mouse normally develops the disease at 8 weeks of age, but here we show that when we inject the animals with the fibrils, we precipitate the disease. So they start showing phenotype at 4 weeks instead of 8 weeks. And in fact, when we looked into the brain, the fibrils co-localize with endogenous HTT protein, and they lower the endogenous levels of HTT. So we're really showing a seeding capacity of the protein. Now, this is important because, and it has clinical implication, because all this time we have believed that Huntington's disease was purely genetic. You needed the genetic mutation in order to develop the disorder. It turns out if you look at a plotting of patients in terms of their CAG repeats, so the mutation, and the age at which they develop the disease, there's a lot of variability. You can have the same CAG repeat and develop the disease at 20. You can develop it at 45, or you can develop it at 85. So there's clearly other components than the genes that regulate the clinical profile of these patients and the variability in the severity that they show in their clinical manifestation. So we actually believe that the extent and efficiency of MHTT to spread will determine if a patient has an early onset with a rapid progression or a late onset with a slower progression. And this is important because not only have I showed you that the protein can propagate in seed, but it's very much expressed outside the CNS, and it's also expressed outside the cellular compartment. We've seen it in the extracellular matrix, we've seen it in the CSF, we've seen it in the plasma. So we're taking advantage of all this information that we found through the study of transplantation to now we've started a collaboration with a group in Austria, and I'm almost, I'm gonna finish with this data. The group is Apheris. It's a biotech company that has developed an antibody against sections of the mutant protein. So what we did is we developed an in vitro assay in which we transfected EC donor cells to synthesize mutant Huntington protein. We noticed that they can release it in the milieu. So then we take this milieu into conditioned media and we expose it to ELA cells, which are the recipient cells. When we inject this into the milieu, the ELA cells are able to pick up some of these aggregates. But when we give the antibody that Apheris has developed, we block completely, almost 100%, the uptake of the mutant Huntington protein. We've gone on to do this in animals, and we see a recovery of some of the behavior in an animal model of Huntington's disease. And in fact, we also see reduction of mutant Huntington protein in the brain of these animals. So I hope that I've convinced you that despite the fact that I'm not showing the potential of cell replacement therapy for Huntington's disease, because I think it's going to be much harder than in the case of Parkinson, where I do believe there's a lot of hope to use this therapy for patients. I hope that I've shown you how much we have learned about the disease mechanisms, and not only that, but that we can actually approach it from a different perspective and perhaps develop antibody-based therapies, for example, which could be used alone or in combination to treat different aspects of the disease. And I think this will be very important in the future to include in the development of new therapeutic strategies. I'd like to thank all my collaborators on this work and all my wonderful students who have really, truly done all the work. Thank you very much.